Hi everybody, I'm Dr. Biomed. Welcome to the Biomed Cafe channel. In this channel, complex biological and medical contents are presented in a simple, classified and summarized form. Also, I have designed a quiz game for you. I will explain about quiz game in the end of this video. The topic of this video is cell junctions. Please subscribe to this channel, and if you like the contents of this video, please like it. Cell-to-cell interaction. Cell-to-cell interaction is the process by which two or more adjacent cells share their microenvironment. In between two or more cells, there are junctions. These junctions act as the bridge between two cells. Now I want to compare cell-to-cell interaction with cell-to-matrix interaction. 1. Cell-to-cell interaction is a communication between cells. But cell-to-matrix interaction is a communication between cells and the extracellular matrix. 2. In cell-to-cell interaction, both communication mediums should be the cells. But in cell-to-matrix interaction, one medium should be the cell, another medium might be a foreign element or any substances. 3. The cell-to-cell interaction is used when a large set of cells are located in the same place. This is being used in the tissues. But the cell-to-matrix interaction is used when the two mediums are placed at the distance. This is used in the immune system. 4. In cell-to-cell interaction, cell junctions are formed in the connection of the cells. But in cell-to-matrix interaction, the cell adhesions occurs mainly to make communication. 5. There is no need to have any receptor in cell-to-cell interaction. But a receptor will be the trigger to start the operation in the cell-to-matrix interaction. Cell-to-cell interaction can be divided into two subgroups: stable interaction and temporary interaction. Stable cell-to-cell interaction. The stable interactions have some different junctions. This type of junction is present in normal growth cells such as epithelial cells. Cells don't have power to break this junction up. They are always in a stable condition. Stable cell-to-cell interaction in plants is Plasmodesmata, but in animals can be divided into three subgroups: tight junctions or occluding junctions or zonular occludence, gap junctions or communicating junctions, and anchoring junctions. Anchoring junctions can be divided into three subgroups: adhering junctions, Desmosomes, and Hemidesmosomes. Plasmodesmata. They are channels passing between the cell walls of adjacent plant cells connecting their cytoplasm. They are located between the two neighboring plant cells. They play a role in facilitating movement of molecules between cells, in linking tissue cells to another, and in making direct contact between adjacent cells. Plasmodesmata have three parts in their structure. The first part is desmotubule. It's a tube of flattened endoplasmic reticulum, or ER, and it runs between two adjacent cells. The second part is plasmodesmatal plasma membrane, and the third part is the cytoplasmic sleeve. It runs between the desmotubule and the membrane. Most of molecules transfer across it. It contains various structures such as actin and myosin that they provide contractile forces to transport. Tight junctions. Tight junctions, also called zonular occludens or occluding junctions. This junction is present between two cells. They seal out the gap between two cells but are selectively permeable. They are located in the lateral membranes of epithelial cells. They play a role in keeping the adjacent cells together, in establishing cell polarity, in allowing the selective diffusion of ions and solutes, and in preventing the leakage of digestive enzymes into bloodstream. Tight junctions have four parts in their structure. The first part is claudins. They form the backbone of tight junction. The second part is occludins. They maintain the barrier between adjacent cells and stabilize the tight junction. The third part is jambs. They seal the intracellular space between two cells. And the last part is zonular occludens, or ZO. They link the tight junction to cytoskeleton. Gap junction. Gap junction, also called communicating junctions or nexuses. They form bridges the cytoplasm of two adjacent cells. They are located in epithelial cells, neurons, cardiac muscle, and smooth muscle. They play a role in coordinating the activity of adjacent cells and in transferring ions, sugar, regulatory proteins, and metabolites. Gap junctions have two half-channel or hemichannels in their structure that are called connexins. Connexins composed of 6 integral membrane protein units that's called connexin. Connexins join together and make the channel. The channel is made up of 12 protein units. Molecules transfer across the channel. Anchoring junctions. They are cell junctions that remain anchored to adjacent cells while being attached to components of the extracellular matrix. They form a tight seal between neighboring cells. They are located in tissues prone to constant mechanical stress, such as skin and heart. Anchoring junctions have two parts in their structure: transmembrane adhesion proteins or linker, such as cadherin and integrin. The cytoskeletal anchor, such as actin and intermediate filaments. They play a role in keeping the cell together, stabilizing the cell position, supporting tissue integrity by holding cells together, and regulating the motility of cells. As I told you some minutes ago, anchoring junctions can be divided into three subgroups: adherence junctions, desmosomes, and hemidesmosomes. Adherence junctions have two attachments. One of these attachments is cell-to-cell attachment. They are located in the lateral membrane of epithelial cells. They have two parts in their structure: transmembrane adhesion protein or linker, that here is cadherin, and cytoskeletal anchor, that here is actin filaments. Another attachment is cell to extracellular matrix or ECM. They are located in the basal surface of fibroblasts. They have two parts in their structure too: transmembrane adhesion protein or linker, integrin, and cytoskeletal anchor, actin filaments. Desmosomes. Desmosomes have cell-to-cell attachment. They are located in the lateral membrane of epithelial cells. They have three parts in their structure: transmembrane adhesion protein or linker, cadherin, cytoskeletal anchor, intermediate filaments, and disc-shaped structure. Intermediate filaments extend from it into the cytoplasm. Hemidesmosomes. Hemidesmosomes have cell to extracellular matrix or ECM attachment. They are located in the basal surface of epithelial cells. They have three parts in their structure too: transmembrane adhesion protein or linker, integrin, cytoskeletal anchor, intermediate filaments, and disc-shaped structure. Intermediate filaments extend from it into the cytoplasm. Temporary cell-to-cell interaction. In temporary interaction, there is no junction created. There is a kind of bond created between the cells. These bonds get degraded and the cell gets distributed. They are not stable and permanent. Its stability depends upon the cell's functions. They are located in the immune system. One example of temporary cell-to-cell interaction is the interaction between the T lymphocyte cell and the epithelium cell of the inner layer of the blood vessel. Thank you for watching. Please subscribe to this channel, and if you like the contents of this video, please like it. Now it's time for the quiz game. I have put the link to the quiz game in the description of this video. Download it for free and enjoy it. In the next video, I will talk about another biomedical topic in a simple, classified, and summarized form. Until the next video, bye!